All right, welcome to your genetics section. Hopefully you guys are very, very excited about genetics. What we need to be looking at is this is from the biology review. This is chapter 12 in our biology review. So you should have at least gone over the concept review and the questions. We're going to be building on that. We're going to be talking about this, and we're going to make sure that we really do understand genetics. Genetics has become much, much more important on the MCAT over the last several years. So it's very, very important that we understand it so that we get those points that we so well deserve for all of our hard work on test day. In this episode, we really only have two learning goals. We're going to trace patterns of genetic inheritance, and we're going to be talking about the chromosomes that we get from our parents, as well as the different types, causes, as well as effects of genetic mutations. Now, as we get into this, you guys know Paul and you know Tyler. You will see comments from them below. You will also be able to ask them questions in the Q&A that is over to the right. So please, please look for what is typed in, ask questions, make sure that you get everything out of this session that you can. Let's get going. Our very first piece here is to really trace the patterns of genetic inheritance. Where does it come from? Now, how many weird special case rules did you guys learn when and had to memorize when you took your genetics classes? How many of those did you have to learn and memorize? Mary says way too many. 987 specifically, a ton. Guess what? Here's the great thing about the AAMC and how much they love you guys. All of these weird, special, funky cases that you had to memorize for your genetics class will not show up on the MCAT. Not in a big capital NOT. They will not show up. Everything that we see on the MCAT is going to follow very basic, very straightforward patterns and rules that we need to know. We just need to know exactly what to pay attention to make sure that we get those points that we deserve. So everyone should be saying, yay, this is very exciting. Thank you, AMC, for not testing those weird rules that my professors tested us on. First thing we have to know is how we are organized. Tell us about chromosomes. How many chromosomes do we have as humans? How many chromosomes? We have 23 different chromosomes. 22 of them are going to be autosomal, and they come in pairs, one from mom, one from dad. And then we have our X and Y, our sex chromosomes. That comes in a pair as well. So we have 23 pairs. We've all seen a karyotype before. A karyotype is a picture of someone's chromosomes. Okay, what do their chromosomes look like? And we put them in these beautiful pairs, mom and dads, etc. 22 pairs of these are autosomes. Okay, everybody has the same basic chromosome 1, chromosome 2, chromosome 3. We have the same genes on those. The other two, however, are sex chromosomes. We're either XY if we're male or XX if we are female. Now, the term that Tyler, Paul, and I want to make sure that you have in your hand is acrocentric chromosomes. What does acro mean? What does acro mean? Does anyone know? Yeah. Acro means kind of close to the head. Okay? Yes, head or top. And Tyler's like, it's really close to a hairstyle. Okay? So basically what that means, okay, Mary, what is acromegaly? You're thinking the right thing. What is acromegaly? Okay, so abnormal growth has to do with the size of the head. So centric chromosomes have something to do with the size of the head. Notice what that means is the centromere is much, much closer to the head, so the P arm is very small. This happens on 13, 14, 15, 21, and 22. Notice on all the other chromosomes, the P arm up here is nice and it's long, pretty. These guys, here is my P arm. It's just that little nub, okay? So that's what we're talking about, is the P arm is just very, very small. All right, let's look at a chromosome in particular. We're going to here look at chromosome number 9. Doesn't matter which chromosome we're looking at. They all have the same relative pattern. And this pattern is a banding pattern, these light and dark bands. Each of these contains many, many genes. Okay, so one light band contains many genes. One dark band contains many genes. But the nice thing is this actually helps us with our karyotypes in lining these up and making sure that we have the right number of chromosomes. When we're looking at these different genes, we have different alleles of genes, okay? Different alleles, one's going to come from mom, one's going to come from dad. These are different forms of a gene. For instance, we could talk the gene for lactase that is persistent into adulthood. As you get older, you will learn that some people start to become lactose intolerant. Some people do not. That's particular for that gene. Another very easy one that we can see a phenotypic trait on very, very easily is eye color. What are the two alleles that the MCAT's going to test us on for eye color? There are two alleles that we're going to be tested on. Dominant and recessive, brown versus blue, right? So when we're looking at this alternate alleles, a brown-eyed allele versus a blue-eyed allele, when we look at these are considered homologous chromosomes, okay? Mom's chromosome 9, dad's chromosome 9, that's where we got these to get our pair of our chromosomes. So when we're looking at these, they have to be homologous. Speaking of homologous and what we can do with them, these homologous chromosomes are separated during meiosis. However, before that occurs, we get crossing over. So here's mom's chromosome 9, dad's chromosome 9. We go through, we replicate them. We have our sister chromatids. We still only have 2 chromosomes. Then we have crossing over. This is where a little bit of mom breaks off and is attached to dad. A little bit of dad breaks off, is attached to mom. One question, what is it called right here where the crossing over occurs? Ah, chiasma. That's my chiasma. Now, these are homologous chromosomes, and we have to know specifically exactly what phase in meiosis does crossing over occur. Done. Prophase 1. Prophase 1, always and only prophase 1. Perfect. Now, as we continue to talk about homologous chromosomes and the different things that we can do with them, once crossing over occurs, the chromosome pairs separate in meiosis 1. Then in meiosis 2, the sister chromatids physically come apart. This is called disjunction. They're coming apart. They're disjoining. If we have non-disjunction, we'll talk about that later and what that means for these gametes and potential offspring. Before we do that, however, we're going to start talking about Mendel's laws. Mendel was a brilliant little monk that played with pea plants. We all know who he is, okay? Mendel's first law was about segregation. What it says is alleles for the same gene segregate during meiosis. This specifically happens in anaphase 1. So they segregate here. So mom's brown-eyed allele went here, and dad's blue-eyed allele went down here. So the alleles for that same gene, they specifically segregate during anaphase 1. All right, and then yes, you are correct, Brandon. he was a little bit of a liar. And here's why he was a little bit of a liar. It's not that he really lied, it's that he didn't quite understand when he was talking about the second law. The second law says that alleles all assort independently. The gametes have each copy, and what it means is that the allele for one thing does not affect allele for another thing. Each allele is separate. But he was somewhat of a liar because what did he not understand? Brandon, since you know he's a liar, what did he not understand? He did not understand linkage. You are correct. He did not realize that these were all on individual chromosomes. However, we know they're on individual chromosomes. So if I have hair color right here and eye color right here, do hair color and eye color separate independently from each other? No. They're on the same chromosome and they're really, really close to each other. So this was the thing that Mendel didn't quite understand. So he kind of lied to us with his second law. We have gotten more clever since Mendel, and we now know alleles sort independently as long as they're not linked. Okay, so we kept his law, we just added a little bit to the end of the sentence with that. Let's look at our first question here. And I want you guys to try this one on your own. So I'm going to pull up a poll here. And I'm going to ask Paul if you could please set us a timer. We're going to give you 45 seconds to do this. I think that's more than enough time for you guys to be able to do this question. Put your answer in, and then we'll go through it. 45 seconds. Good luck. All righty, time is up. And I'm looking at the poll, and I will never display polls because everyone will rush to the one that is clicked the most often. But I will tell you this, 76% of us have this question correct. Very, very well done. So I'll always tell you how many of us got it right. Now, let's look at this. A man inherits two alleles for a given trait. Allele A, the normal allele, is inherited from his father, and the mutated allele A is inherited from his mother. What is the ratio of the man's mature gametes containing the mutated to those containing the norm? So we know that this is going to be a ratio. Here's the easiest way to do this. We can draw ourselves a quick picture. So when we look at this, we see that mom has a lightning bolt. That means that she has a mutated gene. When we go through and we replicate, both of those sister chromatids will have that mutated gene. When we do meiosis 1, the chromosome pairs separate. When we go through meiosis 2, the sister chromatids separate. So I look at this and I see, well, there will be 2 that have the mutation compared to 2 that do not have the mutation. Well, 2 to 2, this is a 1-to-1 ratio. If you need to on test day, you can draw a quick picture like this. It's not going to take you very long. But with more study of genetics, you'll be able to know this right away. Our next concept is dominant versus recessive. All right, we already said dominant and recessive. Dominant alleles show up whether it's homozygous or heterozygous. Recessive alleles, however, have to have both copies, one from mom and from dad. Let's go back to our brown and blue eyes. Which one is dominant? Let's go back to our brown and blue eyes. Which one is dominant? Brown, good. So if you look at someone with brown eyes, you know their phenotype. Do you know their genotype? No, they could be brown brown or brown blue. However, if you look at someone with blue eyes, you see their phenotype. Do you know the genotype of a blue-eyed person? Absolutely. It's got to be blue blue. It's got to be homozygous recessive always. Now, this is an important thing. Star this, circle this, write this down in the brightest color you can find because it's very important for the MCAT. A person who is a carrier cannot express the phenotype. For instance, I have blue eyes. Am I a carrier for the blue-eyed allele? No, I am not. However, if I had a brother, if my mom was blue-eyed and I had a brother that was brown-eyed, he is a carrier because you cannot see it. He cannot have the phenotype. In many cases on the MCAT, this is a disease. So you can't have the disease. You cannot be homozygous to be a carrier. You have to be heterozygous and you do not see the physical manifestation of that trait. Very, very important. We're going to see this in a question in a little while. I can always carry it but pass it on. I cannot carry a brown-eyed allele, but if I had a brother who was brown-eyed, he could carry the blue-eyed allele. Okay, you can only be a carrier. You have the dominant phenotype. You have a single recessive allele. So, for instance, let's look at this. Mom is a homozygous recessive. Dad is a heterozygous carrier. Why am I calling Dad a carrier here? The recessive is green. Why am I calling him a carrier? Because he's yellow, okay, not expressing the green, but he carries a green allele. Nice, you guys got it. So Punnett squares, how many Punnett squares have you done in your life? Bunches. Okay, so you should be used to how this works. Now, when I look at this, my homozygous genotypes have two identical alleles. Mom's going to be green. Heterozygous are different. Now, this is complete dominance because if the yellow allele is there, this is a yellow P, period. No changes. It fully masks the recessive. So of these offspring or potential offspring, how many of them are carriers? Two. Which two are my carriers? My yellow guys. Absolutely. My yellow piece. This is complete dominance. Okay, the yellow completely masks the green. All right, let's look at a question here. And first of all, I noticed numbers, and we're going to talk about those numbers in just a second. A researcher crossed pea plants heterozygous for genes for recessive short and dominant tall. So if they're heterozygous, tell me using T, what is their genotype? Nice. Big T, little t. Perfect. In the next generation, so I've got two of these guys, I'm crossing them. In the next generation, 787 plants were tall. Which of the following answers represents the approximate prediction for the number of short plants in that generation? All right, if we look at this, our parents are both big T, little t, because that's what you said. We look at our offspring, and this is what we possibly have. So the ratio of genotypes is 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. The ratio of the offspring phenotype is recessive, and we're going to see 3 tall, 1 short. Oh, hold the phone. 3 tall, 1 short. That means I should have way more talls than shorts. What answer choice or choices can I immediately get rid of? Oh yeah, D says there are approximately 3 times the number of short plants. No, no. C I can get rid of. Yes. Why? Because it says I have equal number of tall and short plants. Done. What else can I get rid of without ever having to do math? A, is there any chance that if I cross these guys, I will get absolutely no short plants? Nope. I have a 1/4 chance of getting a short plant. Huh. How much math did I have to do here? None. Not any. If I did need to do math, the approximate number of short plants is 1/3 of the tall plants. So I would take my, notice 787, I'm rounding it down to 750 because 750 divided by 3 is easy. So I should have somewhere around 250 short plants. If I did do the math, that's how I would do it. Do I have to do the math? Nope. I didn't need to. Be very clever on test day. Use those critical thinking skills that you are learning from your teachers so that you don't have to waste the time to do the math here. All right. Number three. Number three, I'd like you guys to try as, actually, no, we're going to do number 3, and then we'll do 4 and 5 as homework. So I would like you guys to try number 3. Do your Punnett square really quickly. And when we look at these, first of all, we have a man and a woman who are both heterozygous for an autosomal trait which they do not express. If they are heterozygous for an autosomal trait that they do not express, what does it mean that they don't express it but they're heterozygous? They're carriers. Good. Excellent. They give birth to a son who is also unaffected. What is the likelihood that their son is a carrier? All right. So I'm going to look at this. This is a recessive trait. And I'm going to look at what I have for my possibilities. What is the chance that their son is a carrier? I'm going to pull up a poll. I want you guys to put the answer in the poll really quickly. We know he's a carrier. We know he's a carrier. All right. I will tell you 9% of us, ooh, just went up, just went down, about 9% of us have this answer correct. So what is a carrier genotype? Carrier. Big R, little r. Okay, he cannot express it, but he has to have the allele. Okay, I cannot count which of these genotypes. Which one can my son never, ever, ever be? He can never be little r. Why? Because if he was little r, he would be what? What word would we use right here to describe him? Oh, he would be affected. That is not possible. Never under any circumstances is the homozygous recessive ever considered when you look at carriers. So I have 3 possibilities left. What is the chance that he is a carrier? Two-thirds. Two-thirds. Because 1, 2 out of 3 possibilities of being unaffected is carrier. This question is huge, in importance. This is exactly how it will always work on the MCAT. But now that we understand this, will we ever get it wrong on the MCAT? No. No, we are more clever than that. We can use our critical thinking skills and we can get to where we need to be. Perfect. All right, questions 3 and 4 you're going to do as homework because, again, it's incredibly, incredibly important that you are understanding how to do these questions on your own. Let's look at codominance and incomplete dominance. We know complete dominance says we mask it completely. Okay, our yellow and our green. Masks completely if they have the yellow allele. Codominance is when two things are expressed simultaneously. The big one, the important one that we see all the time on the MCAT is our blood types. So if dad is A and mom is B, notice that we each gave them one O allele. So dad's A, mom's B. They have a daughter, and our daughter is AB blood. Is the A expressed more than the B? No, they are equally expressed. We don't say that this person has AAAB blood because A's are more there, or A plus B blood. We say that it's AB blood. They are expressed exactly the same simultaneously. That's codominance. That's different than incomplete dominance. Incomplete dominance is where the heterozygotes express a halfway in between phenotype. The common one for this is Mendel's pea plants. He crossed a homozygous red with a homozygous white. What color flowers did he get? Yes, he got those beautiful pink flowers. So this is incomplete because it's some red and some white, which made them pink flowers. This is not the same as codominance, okay? Codominance, they're expressed the same. Incomplete dominance, we have a blend of the two different things. And we have lots and lots of homozygous things in human beings, okay? Hair color. You can have something that is a blend of your parents' hair color, okay? So we have lots of things that are blends when we think about them. Oh, absolutely. Height, Brandon, is a perfect one. I am taller than my mom and I am shorter than my father. I am a blend of their heights. That's a great one to look at. All right, let's look at an actual question. In horses, the genes for red coat color and for white coat color are codominant. Okay, so they're both expressed exactly the same. Heterozygous horses have roan-colored coats. Consider a roan-colored colt, the baby, that has a white mother. What must be said about the color of the colt's father? So we see different answer choices in here. So first of all, what is the colt's genotype? He has a roan-colored coat, so he's got to have a red, and we know he got a white from his mom. Yeah, we're going to call it white-red. White-red. Okay. The reason they're both capital is to show that they're codominant. That's why we don't use a capital and a small letter. What was the mother's genotype? White-white. Where did that R allele come from? Had to come from dad. You guys are correct. So what are the possible genotypes of dad? Ah, yes, Nor, take that question mark away. Dad could have either been white-red or red-red. Either way, he could have passed that red on to his son. So what are the potential colors that dad could have been? Roan or red? Notice that thought process led us directly and easily to answer choice C. Love it. All right. Let's look at sex-linked traits. Sex-linked traits are on always and only the X chromosome. Why? Because Y-linked diseases are extremely rare and we will never under any circumstances see them on the MCAT. And X-linked, we're always going to assume is recessive. We do not expect ever to see X-linked dominant. Okay, with that said, we know that males are hemizygous for X. They're XY. So this is a normal, unaffected male. If we have that little subscript, that's an affected male. Females, we have 2 X chromosomes, so we can counteract this. So both of these first two are unaffected. However, the second one is a carrier. Why? Because she's unaffected, but she does have the allele. The only way for a woman to be affected is if she has 2 X chromosomes that have that specific allele. That means it's easier for males to have the trait. Give me some X-linked traits that we see all the time. There are 3 that I can think of that are very, very common. Color blindness, absolutely. Male pattern baldness, and the other one, hemophilia. Those are the 3 that we see over and over again. So color blindness, male pattern baldness, there's a reason we call it male pattern baldness, and hemophilia. Now, does that mean that females never get these 3 things? No, we can get them. It's just a lot more difficult for us to get them. So let's look at what happens. Here, let's call this color blindness. Mom is colorblind. Dad's not colorblind. So we have a carrier female because she's not colorblind, but she's got the allele. She's not colorblind, but she's got the allele. Dad does not have the allele. Dad is not colorblind. All right, so we do our Punnett square here, and here are our possibilities. All right, we can have a carrier female or a normal female. In either case, neither of them is colorblind. We can have a normal male or a colorblind male child. So, males who are colorblind have hemophilia or have male pattern baldness did not get it from their father. They got it from their mother. So the fact that my dad is bald and my brother is bald are completely irrelevant. My brother got his bald gene from my mom, from my grandfather. He didn't get it from my dad because he got a Y chromosome. Okay? It depends. It depends on if mom has that gene. This woman right here has hair. Her son is bald. Okay? So he can still get it. All right, let's go through a quick question here. A trait is discovered that affects an overwhelming preponderance of males. What does it mean if I say overwhelming preponderance of males? What does that mean? A lot, a ton, a high percentage, which means it is what type of inheritance pattern? X-linked recessive. Perfect. Given that the father is showing the trait, dad is bald, and the mother is not, which of the following must be true for their progeny? Okay, I know dad is X affected and Y. Do I know what mom is? I'm going to use my answer choices here to help me. Use your answer choices. If mom was X normal, could any of her children ever be carriers or have the disease? No. Look at the answer choices. The male has it or may have it. The female has it or may have it. Oh, so mom has to carry. Tripty? Nice. Nice. So the answer choices tell me that mom has to be a carrier. Now, what are my possible outcomes for my daughters? Oh, I'm going to fill this out and see what we have. They can be affected or a carrier, not be bald, but be a carrier for it. What are the possible outcomes for sons? Well, they may be affected or they may be normal. Okay, so the males may receive the trait and the females may also receive the trait. I love this. This leads me to one and only one answer choice, which one tells me that either could have it. Yeah, they may have it, they may not have it. Perfect. So here we see females could potentially be bald, but they have to have a bald father and a mother who is a carrier. Okay. Mary, we determined that mom was a carrier because there's nothing in here that if mom was not a carrier, the correct answer choice would have to be males do not receive the trait and females do not receive the trait. Since there's nothing that says both do not receive the trait, she cannot be normal. All right, let's talk about penetrance and expressivity. Mendel was great, but many things that we see are not Mendel's laws, do not follow Mendel's laws. Okay? It's not perfect. Penetrance is the proportion of individuals in a population with a particular genotype that actually express it. For instance, if I have these 5 males, they all have the X that is affected and a Y chromosome, but only 3 of the 5 are affected. So what is their penetrance if 3 out of 5 males who have the genotype are affected? What is my penetrance? Yep, I'm making you do math. 3 out of 5 is 60%. This is 60% penetrance. Now, that's penetrance. If you've got it, you may or may not express it. Expressivity is how much you express it, to what degree. So if I take my 3 affected alleles, 3 affected males, they may not have the same expressivity. One may be severe, one may be moderate, and one may be very, very slight. This is called variable expressivity. They may not always be the same. There is a spectrum of phenotypes here. There's a nice spectrum of phenotypes. All right, know the difference between those two. With that said, we're going to try a little passage here. What we're going to do is I'm going to have Paul set us a timer, and he's going to set us a timer for 1.5 minutes. That should be more than enough time to read and outline these 3 paragraphs. Then we'll jump and we'll look at that outline really quickly. 1.5 minutes to read an outline. Good luck. All righty. So that should have been more than enough time with your phenomenal critical reading and outlining skills. Did everyone get that done? Excellent. So your first paragraph, we know we're talking about cholesterol here, and they give us a normal range. If they give us a normal range, there's a good chance that they're going to give us something outside the norm and ask us about it. So this is cholesterol in healthy humans. Our next paragraph is about HC, that hypercholesteremia, and it gives us a range, moderately affected to severely affected. All right, so this has to do with the expressivity of this. This is going to be, it's moderate to severe. And then finally, it's common in some families and absent in others. So this runs in the family. If it runs in the family and there's no difference between dietary habits, this tells me what about this disease? It's a genetic disease. Absolutely. It's a genetic trait. Yes. We're going to do one question together, and the other two you're going to do as homework. So let's look at this. What do you notice quickly about the answer choices, which are very common on the MCAT? Yes, no, and why? So we can figure out the yes or no, eliminate two of our answer choices, and then the because or the why it is the way that it is. Is it reasonable to conclude that HC is caused by a genetic disorder? Well, we already answered this. Which of my two answer choices are out of here and I'm never even going to consider them again? C and D, because they both say no. Okay, perfect. So a genetic disorder is characterized, as you guys so aptly pointed out, it runs in families and it's regardless of environmental factors. So what we eat doesn't matter if we have this in our family. Now, our passage, what does it say about HC? They said it's relatively common in some families. No significant difference between the dietary habits of individuals in affected and unaffected habits. So we need to know why. All right, this is nice. A says because HC is common in certain families, regardless of the type of diet they consume. You guys are all saying A, you are correct. Do I ever look at B? Nope, I'm done. I'm out of here. I have my point, pat myself on the back, and I move on. The next two you're going to do as homework. Again, you'll get those in the recaps that we give you at the end. It's very important that you use what we learn and do that homework. Now, in our final section, we're going to talk about types, causes, and effects of mutations. So mutations are a change in the structure of the DNA, and they can be for various reasons. Okay, we can have mutagens, we can have carcinogens, we can have different things that happen. So these actually cause mutations, and they can be things that we somewhat do to ourselves, like ionizing radiation if we spend way too much time in the sun, but we all know to wear our sunscreen. Okay, so those X-rays, ultraviolet lights as well. Chemical mutagens, which we expose ourselves to more and more chemical mutagens every day. So these are things that can be free radicals. These are things that are carcinogens. There are other ways that this could happen. We could have transposons. Transposons are DNA segments that can move. Okay, so they're mobile DNA fragments. We also have spontaneous mutation. Sometimes we have problems with our proofreading on our DNA polymerase. Mistakes happen. But we do have good repair mechanisms that exist. They're not perfect, but they're good. Okay, they're there, but they're not perfect. Okay, and we know that because we still exist. All right. Now, let's look at different types of mutations. First things we're going to look at are point mutations. Point mutations come in 3 different flavors, but first let's talk about them. A point mutation occurs when one nucleotide is swapped for another. Say an A is swapped for a G, or a C is swapped for a T. So we're changing that nucleotide. The first thing that can happen is we could have a silent mutation. A silent mutation is where the amino acid doesn't change. For instance, TTA and TTG are both leucine. My protein knows no difference at all. It acts exactly the same. It looks exactly the same. It has no idea that there was a mutation. Does not affect that. These are usually caused by mutations in the third base. Not always, but usually in the third base of the codon. And that's because it's often interchangeable. This has to do with wobble on our tRNA. It also has to do with degeneracy of our code. So both of these code for leucine. My protein, happy, healthy, functioning exactly the way that it should. That's the first type of point mutation. The second is a missense. Missense means that I missed the mark. Okay, it's not horrible, but I missed the mark. Again, we swapped something. Here, we swapped the second one. So we swapped a T for a C. TTA is leucine, TCA is serine. This changes the amino acid, just that one amino acid. This may or may not be horrible. Okay, first of all, if we change something in the first two bases, okay, if we change something in the first two bases, that's less negotiable. That usually changes the amino acid or does something else. Ah, but Hannah's got a good point. It could be a similar type. They could both be hydrophobic or they could both be acidic. If they're similar, typically the mutation is not as drastic. Okay? But it may be depending on where it is. That means that the new amino acid may or may not affect the protein function. Yeah, it could be similar, and it may not affect it too much, or it may not affect it at all, or it could be very, very different, and it could change the way the protein folds, etc. So it just depends on what the change is and where it is. The final one is a nonsense mutation. Ooh, we just made a stop codon. Again, the first and second nucleotides in a codon are less negotiable. Here, if I have a stop codon, this is going to truncate my protein. It's going to be much shorter than it was intended to be. This will typically cause big, big problems. If my protein is only half the size it was supposed to be, it is absolutely, if it functions at all, not going to function the way that it was supposed to. So nonsense mutations, bad mojo. All right, these are point mutations where I'm just changing, replacing one nucleotide for another. The second type of mutation we have to our DNA is a frame shift. This is where I insert or delete a base. This is bad. Okay, if I remove this T here, it changes the entire reading frame from that point forward. Typically after that, we have a stop codon shortly after that. So this arises when we either insert or delete a nucleotide. Changes the reading frame. This is not good. Every amino acid that's there after that is different, and it's going to either alter it or it's going to truncate it. In many cases, there's a stop codon. You guys have heard these before. You know that point mutation comes in 3 flavors. Okay, frame shift comes in 2 flavors, insertion or deletion, and they're bad. We don't like frame shift mutations. All right, this is individual pieces or individual nucleotides, individual genes. We can also have chromosomal mutations. This is where we affect large parts of our chromosome. So some can involve a single chromosome. For instance, I can delete a chunk of the chromosome. Well, I just lost those genes. So that chromosome is now shorter. I could duplicate part of that chromosome. Well, now I have multiple copies of those genes. This is going to potentially change things. And the final one is an inversion. I may take a section of my chromosome, completely flip it over. It changes the order of the genes. Now, this doesn't tell us the severity of what has happened. This is just a mechanism by which our chromosomes can change. Okay? But it doesn't tell us the severity of what the change is. Depends on what is lost or what is inverted or what is duplicated. Another type is multiple chromosomes, where we have different chromosomes that do things that are weird. For instance, if I have these two chromosomes, notice they're not the same chromosome. Okay? I'm crossing one chromosome to another. When I do this, if I have that insertion, the yellow chromosome is now shorter. The gray chromosome is now longer. So DNA moves from one chromosome to another. We can also have a translocation, where the pieces just switch. Again, if I'm switching these or I'm moving them, this is a mechanism of action. Does it tell me how bad the change is? This is just a mechanism of action. Does it tell me how bad the changes are? No, not at all. Okay, so no, Mary, we can't say there's always a bad result. Okay, these are mechanisms of action. All right, genetic mutations, we said, can be advantageous. Yay. Okay, for instance, heterozygotes for sickle cell anemia are healthy. They don't have the disease, and they're malaria resistant. There can be deleterious or bad mutations. Okay, we know different ones. There can be loss of function mutations, so that cannot carry out its normal mutation. Okay, so heterozygotes may have an intermediate effect here. We could have cystic fibrosis. This is a good example of that. We could have a gain of function mutations. So we now have new mutations. Some can be good, some can be bad. Okay, Huntington's disease is caused by a modified protein. Some gouts are caused by overactive uric acid. So depending upon what we can have different types of things going on. Always remember that. Now, we're going to look at the passage for passage 2 really quickly together. In humans, chromosome X is a protein that induces normal cells to emit more calcium. Excellent. To examine the mode of action, a biologist screened thousands of families with hormone X deficiencies. The screen turned up 2 unrelated families in which many males but no females had the defect. Ooh. So this first paragraph tells me we have a defect, and it is sex-linked. Then I have this nice list of things. In both families, we know that there are problems. In family 1, protein R was smaller. Okay, so I'm going to write that down. Family 1, smaller protein R. Family 2, when we look at family 2 here, we couldn't phosphorylate protein P. So if protein P cannot be phosphorylated, is the problem with protein R or with protein P? Protein P, because it can't be phosphorylated. So protein R is the problem in family 1, it's teeny. Protein P is the problem in family 2, it cannot be phosphorylated. Many things, if they're not phosphorylated, they are not active. Okay, if I need to go back to this number list, I can. But I'm not going to spend too much time on it. Let's look at question 11. Based on the passage, the mutation in the DNA in affected males from family 2 most likely results in. What is the problem with family 2? Well, we said their protein P cannot be phosphorylated, which you guys said that means there must be a problem with protein P. Okay, which one tells me about that? Well, first of all, it has nothing to do with protein R. So I look at my answer choices. A is out since it says protein R. C is out since it says protein R. And then I look at B or D. B says the replacement of a single amino acid in protein P. Could that cause a problem? Sure it can. Do I look at D? No. All right. Questions 11 and 12, you're going to 12, 13, and 14, you're going to do as homework. You will have those in the review that we give you, the recaps. So our final thing we need to look at is genetic inheritance. It comes from both parents. They have different types of things. We have dominant, we have recessive. Genetic mutations may be good, may be bad. Can occur chromosome or nucleotide level. We have point mutations and we have frame shift mutations. All right, hopefully you feel a lot better about genetics. Thank you so much for joining us. We will see you in the future. Have a phenomenal night. Bye-bye.